Thank you, Dave. So, hello everyone. As Dave already said, today we are gonna have our second session of the computational sessions, and we'll have this session totally dedicated for QTL analysis, and we will start with Maria Caraça Harter. Maria will begin the session talking about GWAS Poly and dia-QTL. I think you split in two sessions, right, Maria?
Yes, I will talk first about GWAS Poly and then dia-QTL.
All right. So, Maria, if you are ready, you can start your first session. And everyone, just reminding you, if you have any questions, you can drop a question in the chat and we will read. Or you can just open up your mic and ask your questions. Okay, so Maria, the
Yes.
floor is yours.
Thank you, Gabriel. Yes, please, if you see any question on the chat, you can stop me at any moment and I will stop for answering the questions as I go.
Okay.
All right. You can see my screen?
Yes.
Great. So, let me close this. Right. So good morning or afternoon, everyone. I'm Maria Caraça Harter. I'm a researcher at the University of Wisconsin. I work in the Jeffrey Endelman Lab. And today I will be talking about two different programs that we use for QTL analysis, GWAS Poly and dia-QTL. So this tutorial will be very explanatory about how we're going to use both of these methods. In the last year, we have specific examples for each of them. And I thought it was very beneficial, but throughout the last year, there have been some questions, and I was trying to address some of these questions and perhaps some solutions for you to explore. So I have shared a link in the chat. For anybody who's just joining, please let Gabriel know and he can share it again. You can see the link, Gabriel, right?
Yes, I just posted the link again in case anyone missed it. Yeah.
Wonderful. All right. So we will start with GWAS Poly. GWAS Poly is a package that has been designed for genome-wide association studies in autopolyploids, but also works for other ploids, for diploids. So this package has a vignette that is very well explained, that is available on their website. And this illustrates the basics workflow of using a potato data set, because we have used potato data for this analysis, and that's what I will be touching on since I work in a potato breeding lab. So just so you know, the basic workflow of GWAS Poly, we use these main functions. The first one is the read GWAS Poly, which will read the marker and the phenotype data. Then we have the set K parameter, which will specify the covariance matrix for the polygenic effect. We can also set the parameters for additional models, such as environments and other covariates. And the main function, the GWAS Poly, is the one that will perform the mixed model for the association test. We can also check the inflation of these p-values by using the QQ plot. Then we can establish a threshold and be able to visualize these associations by plotting the Manhattan plots. This package also offers the option for getting the QTLs, and it will identify them by chromosome, as well as feeding specific models where we will compute the R-square to see how much variance is explained by each of the QTLs that are identified. And finally, you are able to write the whole output to one specific file, and this is optional. So for this package, I have made a website on GitHub, and where you can download the data sets for running these, and there is a tutorial. So the link that Gabriel just shared with you will lead you to my GitHub page, and that is what I'm showing you now. On this GitHub page, there is 3 main folders: data, images, and tutorial. In data, I have compiled all the data sets that we will be using. I will describe them in the next slide. Then the images that have been generated by following the tutorial and the tutorial. So if you would like to follow along with me, all the analysis that I'm going to explain, you can click here on code and download zip. And when you download zip, you will download all the data sets and everything that is included on the website. And you should be able to follow along with the script. Can you see that on the screen?
Yes.
Okay, perfect. So for anybody that is already on the link, if you scroll down here, there is another link that says tutorial link. And if you would like to see the results from running this script, then you can just follow in this website. And I will be explaining all the outputs in here. So going back to my presentation, excuse me. The data set that we're using for this example has been published in this paper, The Genetic Architecture of Vine and Skin Maturity in Tetraploid Potatoes. This data set has data for 2 different years for tetraploid potatoes to analyze or to identify QTLs for vine maturity. Last year, this is the only data set that I use for the example, but what the questions that we usually get when people are emailing us about GWAS Poly is the possibility of analyzing or identifying QTLs when there is mixed ploidies, meaning that some of the individuals in our data set will be diploids and some will be tetraploids. So for this session, I have added data that was published on this second paper using haplotype and QTL analysis to fix favorable alleles in diploid potato breeding. In potato breeding now, there is the new techniques that we're using for developing new diploid breeding lines. Therefore, now some of our data sets will have a combination of tetraploids and diploids. And it's very important for us to be able to analyze these together. So what I have done for this session is combine the data sets for both of these papers in order to identify the QTLs. And depending on what crop you're working on, definitely this is important because as in any other breeding program, many of the lines are not always in the same ploidy, and we should be able to have something available to analyze these together to identify some QTLs. So the first part of GWAS Poly is to use genotype calls. There is different methods for assign the genotype calls for polyploids. Some of them have been explored in the previous session. The one that I'm going to touch on in this opportunity is Polybreeder. Polybreeder is an R package that was designed to facilitate the use of genome-wide markers for breeding autotetraploids species. This package will assign the tetraploid genotype calls from a SNP array. So all the data that I will talk today about is on SNP array data, but there is also a possibility to do this for sequencing data. It will use the function genocall, which will make the genotype calls for each allele ratio from the data matrix. So using polybreather, as you can see on this figure, we will be able to make the genotype calls for the 5 different genotypes that you can find in tetraploids. And this dosage will go from 0 to 4 for allele number B. So you will have the ratios of X and Y, and when you plot them, this package will be able to classify them into the 5 different genotype calls. And that's what we usually see in tetraploids. So, but like I said, in tetraploids, we have 5 different genotype calls, but that's not what we see in diploids. And if we're looking into mixing these data sets, we need to also be careful about how we're going to be combining this data. So what we see on diploids genotypes is only 3 groups, because the 2 groups from here will not exist. Since they are just diploids, we will have dosage 0, 1, and 2 instead of 0, 1, 2, 3, and 4. When these genotypes have been assigned, if we just combine these data sets, then we will have dosages of 0, 1, and 2 for the diploids. And these will not represent what we're really trying to evaluate, because the tetraploids, the heterozygous, excuse me, are here on the group number 2. And here, the heterozygous are on group number 1. Therefore, if we just use these dosages, when we combine these data sets, we will be only telling our model that we have these 3 groups of dosage, which is not true. So there is a very important step beforehand where we will have to recode our data coming from the diploids. And these dosages, instead of being 0, 1, and 2, will need to change to be 0, 2, and 4. In that way, we will have the homozygous for allele A and B, just the same as on the tetraploids, and the heterozygous, meaning that they have 1 copy of A and 1 copy of B, will be the same heterozygous as the tetraploids. So this is a very important step before we merge our data sets to do the analysis. So for anybody following this tutorial, the first part under GWAS Poly was to do this data adjustment from diploid to tetraploid genotypes calls. And here you can see that I am going to convert the data coding from 0, 1, and 2 to 0, 2, and 4. So this is the file that you will see inside everything that I asked you to download, the file for the diploids and the file for the tetraploids that are initially separated. But eventually I merged them first by recoding. All the individuals that have 0 will be still 0. The individuals with have genotype called number 2 will be 4, and number 1 will be 2. In that way, when you look at one specific individual, you can see that it will change from having genotype 0, 1, and 2 to have genotype 0, 2, and 4. After this, we have made these changes that we can merge both of these data sets. And the code right here is what is going to be merging these 2 data sets and generating a new genotype call file that we will use later on in GWAS Poly. So I'm just trying to explain you what is happening at each step. You're very welcome to follow along. This should not take time. And if you open the script, I just wanted to show you where you can open the script. So when you go to tutorial, in tutorial, there is 2 files. If you open this RMD, then it will take you to, if you have already downloaded RStudio, it will take you here. And here is where you can run all the lines that you can see on my GitHub website. Okay, so once we have combined these genotypic data, then we can go to the next step in GWAS poly analysis, which is reading the data. Okay. So this function, read GWAS poly, will ask you to first determine diploidy, which in our case will be 4. Even though there are some diploid lines, you will just put 4 here. Then you have the phenotypic data. The phenotypic data, the genotypic data, and the number of traits that for this example is only 1, which is by maturity. And then you also need to determine the format of your trait. Here you pretty much only pass the names of the files as strings for both the pheno and geno file. And you can also use BCF files for anybody who has that type of data. If you go to the vignette on the website of GWAS Poly, you can see how to upload such type of files. So for this example, we have a total of 8,200 SNP markers, and combining both of these data sets from both of these papers, we have 866 individuals. The second step is to go for QTL discovery, but there is few steps in between. The first one is to use the setk function, which will account for population structure. And this is a K model that you can determine based on what are the parameters that you would like to use. For instance, the newly available local LOCO method will leave one chromosome out for computational efficiency. And there will also generate a different covariance matrix that will be calculated for each chromosome based on the markers from all other chromosomes. So when you put LOCO equal true, this is what it's going to do in your analysis, and it usually will be, it will take less time. And the other option is that you can also change the number of cores based on the computer that you have available. So then this will also save you some time. The second part is to determine the parameters. And here you will include the covariance in your model. For instance, one of the factors in my model is environment, because 2 years were evaluated for the tetraploid potatoes and only 1 year for the diploids. So I'm using that data as a factor, and that is called environment. The only important thing here is to remove markers that are below the frequency threshold. It is recommended to use 1 minus 5 divided by the number of individuals. So like I said, in this data set, we have 866. But depending on how many individuals you have in your data set, you will change this number. And the last part will be to run the function GWAS poly, which will conduct a hypothesis test for each of the markers that you're analyzing. And the traditional GWAS model assumes an additive relationship between trait and marker allele dosage. But GWAS poly can also test non-additive models, which you can find in polyploids, such as dominance models for 1 or 2 of the genotypes. And that's very important for some traits. For this example, I'm just using an additive model. And the trait that I have that is of interest for this study will be band maturity. For the parameters, we will use the parameters that we said before, and the number of cores too that can be adjusted. once we have that, we can also set a significant threshold. This is also a difference from the previous workshop from last year, because now there is more methods for establishing this threshold. So this threshold will control the genome-wide false positive rate. The original Bonferroni method, which is very widely used, excuse me, the overall significance level is divided by the number of tested markers. However, this is usually very conservative because the tests are not independent due to the LD. So the LD between markers can also be used to estimate this threshold by using the method, this method MF, which is accounting for the effective number of independent tests. There is other methods that you could use too. So it's the Bonferroni, this effective number of independent tests, or permutations. In some cases, some studies also for publication will require to have close to 1,000 permutations, so you can adjust this in here as well to set the thresholds, which in our case here was determined as 4.48. So then the next function, the Manhattan plot function, will allow us to see these Manhattan plots. Here you can determine which model you want to look, because by default, if you are evaluating more than one model, all the models will be plotted together. But it also allows you to plot them separately. And
Maria,
yes?
just one thing, there are two questions in the chat. Would you
Okay.
like to answer them now or at the end of the session?
Oh, no, I can go on now.
Okay, so I will read them to you. So the first one is, do you filter the markers that do not follow the expected segregation? And I think this is in the input that step.
What do you mean by the expected segregation? In diploids, there is only 3 genotype calls, and in tetraploids, there is 5.
Yeah, there is no clarification. If you wanna clarify this question, Paula, feel free to open up your mic.
So you have a minor allele frequency that you can remove markers that are not common and things like that. Yes.
Mm-hmm.
Yeah, I hope that answered your question, but if not, please just follow up with another question, Paula.
There shouldn't be any segregation distortion measurements because in GWAS, we're normally looking at a diversity panel instead of F1, F2,
Oh,
other
yes.
mapping populations. We can use segregation tests then. So hope that answers the question.
Great.
Yes, this is a diversity panel. There is not one specific family.
Mm-hmm,
We're
mm-hmm.
going to use specific families for QTL, but not for GWAS poly.
Yeah, great. The second question is from Sarah. She's asking what other phenotypic data format you can use, for example, classes or binomial presence or absence.
Yes, you can use binary traits and you can use quantitative traits. It will work for both. This is by maturity, so it is quantitative trait, but I have used the same methods for presence of absence of color on the skin, and it works pretty well as well. The way you denote the trait is different. If I'm not wrong, it's either Y or N. And if you look at the link for GWAS poly, it will tell you how to code a binary trait.
Nice, thank you, Maria. And there is a last question here. Are we allowed to cheat on the data set for a 4x crop for which the calls were made as a diploid? Is there a way? So I think the question is that if you have like a genotypic data set that was called as a diploid, can you cheat in any way to treat it as a four tetraploid?
As a tetraploid?
Yeah.
If you only have 3 types of genotype calls, 0, 1, and 2, or 0, 2, and 4, I think if you put that it's a tetraploid, might lead to error because it might look for the other genotype calls. But what would be your reason for trying to cheat on it? You could also use ploid equal to if you know that you only have 3 genotype calls.
Mm-hmm. Yeah.
The GWAS poly works for diploids as well. So
Yeah,
I think you should just put 2.
Great. Yeah, I guess the situation where that could happen is that if you have a true tetraploid species, but the data was called as a diploid, if there is a way you could use it, but yeah, I think you are losing information because you are collapsing all the heteros in one class, right?
Before, when there was no availability of GWAS analysis for polyploids, that's what people used to do. They will diploidize the data, but you are missing information.
Great. And while you were answering those questions, there is another one. So based on your experience, is it okay to assume that the same allele dosage effect for a target trait in 2X and 4X individuals?
The same allele dosage effect. So you have to be, so for these, for instance, for this example I'm talking about, I have used the same array and the same SNPs for both populations, the diploid and the tetraploid. When you do the genotype calls, yes, you have to determine which is allele A and allele B. But since it is the same data set and you are also using actually the same function to do the genotype calls, I think that you can work with them together. So it's safe to assume that only on the small subset of individuals that are diploid, you will only find 3 different genotype calls instead of the 5. So pretty much you're combining them because you can trust that some of the samples can be diploid, even though you're calling them on the model as tetraploids.
Great. All right. Thank you very much, Maria.
Okay.
I think all the questions were answered.
Great. So the most significant markers will be detected here. Here, and for this example, for bimaturity, there was a peak on chromosome 5, which is where the maturity gene is located, the CDF1 gene in potato. So it worked pretty well combining both of these ploidies. I know this is a question that has always come back to us on GitHub when people are looking for some help in GWAS poly, and definitely this is what we have been telling people to do, and it has worked so far. I know that there are many other crops where you also have mixed ploidies, and if you're using the same SNP array to analyze all the populations, this is one way to get it all together. So the other important function for GWAS poly is that you can get the QTL effects and the R squares. So by running this function, get QTL, then you also determine which model you want to analyze and the base pair window, because you can have, based on the linkage disequilibrium, how many of the markers it will tell you. If you don't determine this, it will just give you all the list of the markers that pass the threshold. And many of the potato SNP array markers are not haplotype specific, therefore the LD is quite low, even at 0th instance. It's because there's highly heterozygous. So the base pair window will prune out to return only the most significant markers within this specific window size. And finally, you can also feed QTLs to see, for instance, in this specific case, we have a QTL on chromosome 5 and one on chromosome 6. So you could analyze also if there is epistasis between these two markers that are on the peak of each of the QTLs. And that's another good function that we have using feed QTL. Here also the important part is that you have to specify again that you're using environment as a factor in your model to analyze whether or not it has there is an epistasis between these two. And it will tell you from the R-square how much of the variance is explained by the QTL, which for instance, in this case, the QTL on chromosome 5 explains 24% of the variation and the QTL on chromosome 2 only 2.4. And yeah, the time is flying. I think already we passed the 30 minutes for GWAS poly. So I guess I will check again if there's any other question about GWAS poly before I go into DiaQTL. Are we good?
No. If you have any questions, please feel free to drop in the chat before Maria starts the other section.
Can I ask one question?
Yes, sure, go ahead.
If I have a multi-generational data, several generations, can I use it to do that, to do the QTL across generations?
What do you mean by several generation data? Do you have like F1s, F2s, F3s? Was it in breeding or?
Well, I'm working with polyploids, so that will be generation 1, then you select parents, you make crosses, you have generation 2.
So
and
if
so
you have like JK was mentioning, if you have a diversity panel, which means several crosses, some of them have some relatedness, some of them don't. Yes, that's exactly what GWAS poly is for just a diversity panel. It doesn't have to have a specific population structure where you, for instance, I will be explaining on DiaQTL when you have specific populations that have the genotypes also from the parents and it's different. In GWAS poly is more open to have a diversity panel and you will account for the structure in the model. So it should work.
All right, thank you.
Okay, so I
Sorry.
guess,
Yeah, there's one question in the chat.
okay.
Is there any way to figure out which model is better between additive and non-additive?
Yes, so when you want to, you can compare the AIC values for additive and 1 dominant, 2 dominant models. If you go to in my script here, if you go to GWAS poly in the vignette, this vignette will show you how to compare AIC values and determine, and then you should be able to determine which model is better for you. So you just compare the AIC values. Go to the vignette of GWAS poly and it will show you there exactly what to do.
Thank you, Maria.
All right.
There's another one. Is it possible to do multi-alleles GWAS analysis?
So it depends on your genotypic data. What the data I'm using right now is B allelic because it's an SNP array. So you only are considering allele A and B. If you have BCF files or data from sequencing, I will assume that it will also be biallelic. So I am not sure about multi-alleles. When you have haplotypes, then it's different. But for GWAS poly, this is biallelic data. And if you think about it, when you talk about dominance and these things, it will be assuming that it's only 2 alleles per marker.
Got it.
Um,
I don't know if we have more time. There's one more question here in the chat, but
yeah, I might have very limited time for QTL, so maybe we can keep all the questions by the end of the sessions, and then I can try to go back to it.
okay.
Will that work?
Yes, sounds good.
Okay, perfect. Great. So now we're moving into QTL. So the QTL package is an R package designed for QTL and haplotype analysis in autotetraploids and also in diploids for diallel populations. So the phenotypes will be regressed on the genotype probabilities, and these regression coefficients are random effects. So here there's two main things. First, we need to have a diallel population, meaning that these are populations that are interconnected by the parents. And second is that we use genotype probabilities, not genotype calls. So we will have to calculate these genotype probabilities before we can use QTL. There is few ways to calculate these genotype probabilities. The software that I'm going to be explaining about is Polyorigin, which means is in this case is recommended to calculate these genotype probabilities in tetraploids when you have more than one population. There is other softwares that have been described in the previous sections, but as far as I'm aware of, they do not do it for multi-parent populations, only for biparental populations. So for the first part that I said, for the genotype probabilities, we're going to use Polyorigin, which will perform a haplotype reconstruction. So this package has been developed to run in Julia. And for anybody that has been part of this workshop before, I have explained this in the previous workshop. You can watch that video too. I believe that it is uploaded on YouTube. And I know it sounds a little complicated to run it in a different program, which is Julia, but it is very straightforward. And all the files that you need to run this example are also in the same folder that I asked you to download. So you have all the material. There is vignettes that are available and that are also very helpful. And you can follow that along as well if you would like. So in Julia, we're going to run this code, which is using the Polyorigin package. We'll call it package. And for these, there is three main files that you will need. The genotypic files and the pedigree files to start with. The pedigree, how are these files looking? So first of all, like I said, Polyorigin allows you to do interconnected populations. So for instance, we have three different parents in this data set. And these three parents have had crosses in between them, and each of their population is for over 100 individuals. It depends on the ploidy that you're using, how many individuals you will need to determine the haplotypes. For instance, for diploids, what is much easier to track the haplotypes, you will not need as many individuals to have these interconnected populations. But for tetraploids, you need a larger number of individuals. So then you can really rely on these haplotype calls. And so this is called a diallel population. And then to run Polyorigin, you will need to specify the pedigree file. You need to determine whether or not you have a physical map. So for the markers, you can have either the physical or the genetic distance. And if you don't have the genetic distance, you can have the physical distance, but you need to determine the recombination rate, which for potatoes is widely used as 1.25. It will depend on your crop. Then you can also determine whether or not you want a refined map or an order, if you want to recalculate the order of the markers. You can also just have the analysis of a chromosome subset in case you're just interested in one specific chromosome, if you know what the trait, if there is a gene that is controlling the trait of your interest. Because this definitely takes a longer time. So it will take on around 8 minutes just to run one chromosome. It will take, I think, about 2.5 hours to run for the 12 chromosomes in potatoes. So that I do not want you to run these right now while we are in the presentation. You can try that later. And if you have any problem, please just reach to me, send me an email. I will try to help you. So what are the outputs from polyorigin? It's mainly these 7 outputs where we will have the genome probabilities, the map refined in case you have requested to have it, the parent's phase and the correction of them, and the polyancestry, which is the file that we really need for using QTL. That's the file that we need. And another function that polyorigin has is to visualize the haplotype polyancestry of the polyancestry file. So for each of the individuals, like in this example, I just have chromosome 1 and chromosome 2. You, in this haplotype map, you are able to identify their combination events and which haplotypes are carried for each of the parents. So you have here on this axis 8 different haplotypes because these are tetraploids. You have 4 haplotypes that can come from the mother and 4 from the father. And you can see where there was a recombination event and there was a switch from one haplotype to the other. And yeah, so that is the data that you need before you go into the QTL analysis and QTL. So that was the haplotype reconstruction. And then once you have that polyancestry file, you need 3 input files that are needed for QTL analysis: the pedigree file, the genotype file, which are the genotype probabilities, meaning the haplotypes, and the phenotype file. And QTL, the good part of QTL is that it has several functions to prepare the input files from the output from several linkage analysis packages. Like I said, some of these have been described in the previous session. For haplotype construction, you can use polyorigin, mappoly, rabbit, or OneMap. And QTL has a function for converting the outputs of each of these different packages into the files that you will need to run QTL. So that is very, very helpful, and you will not have to deal with rearranging things or anything. It knows how to work with the outputs from other softwares. First, you're going to you will need to download these packages beforehand. And once you call the packages, you will set the working directory to where you have your polyancestry file. And the files that will be generated will be saved in the same working directory. So once you run this function, convert polyorigin, which for the other softwares will be, for instance, convert matpoly or convert rabbit or OneMap, then it will automatically generate 3 files, which are the 3 files that you need to input for the QTL analysis: the genome file, parents, and pet file. Then the read data function will read all these files. And here also you need to determine the ploidy of your crop, which in this case is tetraploid, and the dominance model, because it could be from additive up to dominance equal 4, which is the largest of the ploidy. And you can also determine the number of cores depending on your computer and what you have available. So for this example of QTL, there is 394 individuals that have pedigree and genotype data, and they also have the phenotypic data. Then the next function is scan1 for QTL discovery, So this will perform a hypothesis test for each marker bin, and by default, the additive model is the one that is used when dominance is equal to 1, which means the predictor variables are found their haplotypes dosages. So depending on which haplotype you have is how that would be affecting this specific trait. Here you can also determine, excuse me, the chromosome. If you put null, it will analyze all the chromosomes. Otherwise, if you want to save some time, you can just put the chromosome of your interest because you're doing fine mapping. And yes, so it is very straightforward. After you perform that, you can also determine the detection threshold. The genome size will need to be used to determine this. So to get the genome size of, in this case, potatoes, we will use the function getMap, and this will use the chromosome and the centimorgans to determine what is the genome size, which in this case is 13.12. It's pretty close, around 13 for what is for potatoes. It will give you more detailed number regarding of the population you're using. And the next one is to use the DIC threshold function, which will allow you to determine this threshold based on the genome size. So you need to determine here the genome size, the number of parents, because some populations will have more parents or some populations have less parents. So that will also determine your threshold, be a factor in your threshold. The ploidy, which in this case is 4, and the alpha, if you want a 95% alpha or a 90%, depending on what threshold you would like to use in your study. So for our example, the alpha 95% is 17.74, and it's a little higher when it's 90. So then once you have the threshold determined, you will use the scan1 summary function to return which markers have the highest DIC scores on each of the chromosomes, and it will plot it with a profile where you can also determine your threshold. So it's using functions from the ggplot2, and pretty much you will just draw an intercept where you will put the threshold that you previously determined. And yes, you just put a scan1 summary, and then from that element, you just put plot, and you can see that again, we have the same, this is also for body maturity. So we have the same QTL on chromosome 5 for the CDF1 gene and potato. But there is also another QTL on chromosome 4, smaller. So for the QTL discovery, the peak on chromosome 5 and the 16 centimorgans, like I said, coincides with the CDF1 gene. And there is another QTL on chromosome 4 too that was significant, so that was important to look at afterwards. For QTL modeling, you will be able to, after the discovery of this QTL, is to feed a multiple QTL model. And so in this specific case, since you have 2 QTLs, you will determine this on QTL data frame, where you put the marker names for QTL and the chromosome 4 and chromosome 5. And you can also have different dominance for both of them. Perhaps one of them is additive and the other one is digenic dominance or trigenic dominance or quadrigenic dominance. Again, if you want to determine which dominance is more significant, you just compare the DIC values. So this is important for multiple QTL models, which is commonly seen in other traits where more than one gene is contributing to it. So that's another important function in QTL. For QTL modeling, you can plot the additive and the genic effects for each marker. And here, in this specific bar plot, how do you differentiate the haplotype effect? Is, for instance, in this example, you have 3 parents, parent A, B, and C. And then you have 4 haplotypes per parent because these are tetraploids. So how you interpret this is that, for instance, the bars that are above 0 means that they have a positive effect on biomaturity, and these have a negative effect on biomaturity, meaning early maturity and late maturity. So you can identify which haplotypes confer, what is the effect on biomaturity of each haplotype, and based on that, you can determine which haplotypes are the desirable, or if you have a breeding program, which individuals are carrying the haplotypes that are of your interest, and just keep those for recurrent parent selection or for continuing your breeding program. So for the QTL modeling, you will look at the variation too. So this will tell you the proportion of the variance of each of the effects. And this is specific, you have 2 QTLs on chromosome 4 and 5, and the residuals. If you have also a polygenic effect, you can include that in the model for feed QTL, and then you will obtain also the proportion of variance that is attributed to the polygenic effect. And once you know the effect of each of the haplotypes, you can also have some haplotype-based selection, meaning that using the function haploget, you will have the data set that is the output from the IQTL, and the marker that is, for instance, this the pig marker on chromosome 5. And here, this element will have all the haplotypes at least a specific marker. And then you can ask which individuals have this haplotype specifically that to me is giving me early maturity, which is desirable in potato breeding. So then when this value is above 1, that means that these individuals have 2 copies of this specific haplotype that we want. And for our data set, there is these 5 individuals. I don't know why it's moving so much. There is these 5 individuals that carry 2 copies of this haplotype. But how that is rare, and why is it that so appealing for us to use polyorigin? It's because this package also allows you to identify double reduction, which happens in potatoes. It does not happen in every polyploid, but it does happen often in potatoes, meaning that there you can inherit instead of 2 haplotypes from one of your parents, there is a double reduction event here, meaning that you can have 2 copies of this haplotype and just carry that. So that is something that happens in potatoes. It's not very common to see that in other crops, but it's very important for us that there is a software that accounts for that, and therefore we're able to identify individuals that have the double reduction on the haplotypes perhaps that we are interested in, which in this case is for biomaturity. So in that way, you can also make some selection based on the haplotype effects. And I guess that concludes the QTL, and I can get some questions. I know there might be some questions in the chat, and we have a few minutes to go over it.
Maria, there are some
Yes.
questions here. Let me start. So, how do you determine number of haplotypes? Is it one haplotype with copies or multi-haplotypes, each one copy?
So when you have a tetraploid, that means that your mother and your father, each of them, because they have 4 copies of each chromosome, you can have 4 different haplotypes coming from each of them. So it depends on your ploidy how many haplotypes you can have and how to map it and track them back. Because the importance of this is to know, okay, so you have 4 haplotypes from your mother, 4 haplotypes from your father. and you only inherit 2 haplotypes from each of them, and then they get combined, and then you're a tetraploid, right? You have 2 haplotypes from your mom and 2 haplotypes from the father. So to identify which haplotypes you have from the parent, that's what is giving you that information. That's what you're tracking back. And the haplotype effects is from the parents. So in this haplotype effect, this is each parent. There are 4 haplotypes and the effect on this trait. I don't. Does that solve the question?
Yeah, I think so. There's another question from Sarah. She's asking if the parameter recombination rate is necessary to run polyorigin.
If you don't have the genetic distance, yes. If you only have the physical distance, you need it.
Great. There was another question. I think it was regarding the previous presentation on GWAS. The question is it possible to use GWAS in a combined population of 4 biparental mapping populations with one common parent across the 4 populations? And then Jikin answered that it was probably better to use dia-QTL. Is that right in this situation?
Yes, if you have if they all have a parent in common, definitely it's better to do the haplotype map because you have the information needed for that.
Mhm.
You will take advantage of it to track back the haplotype effects.
Uh-huh. And in the same thread, so after Jikin answered the question, there was another question. If you have any recommendations for tetraploids with a degree of preferential pairing.
Preferential pairing. So that means that you will be having to know that you will need to have a diallel example, right?
Mhm.
You will need to have a specific population. So again, I think I will take advantage of using dia-QTL for preferential pairing is because you have an F1 from 2 parents. And polyorigin can do multi-parental populations, but can also do a biparental population. So you can just use one population at a time too.
Mhm, mhm. There's another one. I think it's a yeah, I think it's a question about the definition. In your definition, haplotype means homologs, while haplotype may mean gene alleles.
So yes, I don't have it here, but yes and no. For instance, if you have a biallelic gene that, let's imagine CDF is just biallelic, you only have 2 alleles. One allele confers early maturity, the other allele confers late maturity. If you look at this haplotype map, you could say this is allele A, allele number 1, and this is allele number 2, because based on the effect, you can identify them, right? But that's the case for some of resistant genes, when then you have a susceptibility and resistant alleles. But it's not the case for many other traits, such as, for instance, this specific gene, which is CDF1. There is 4 different alleles that have been described for this gene that have been studied before. One is the wild type, which confers late maturity, and there is 3 alleles that confer early maturity. So all the early maturity haplotypes could be either of the 3 alleles. And it will be hard for me to determine from this plot which specific allele is each haplotype. Does that make sense?
Mhm. Yeah. And I don't think we missed any other question. Let me just check again. Okay. Yeah, I think all questions were answered.
All right. Yeah, I think it's one already. Anyways, I
Yeah.
don't want to take your time.
Yeah, no problem. So,
I think I have one last one.
yes, sir. Go ahead.
Is there a sequencing technique that allows you to make the calls for a tetraploid population?
Do you mean?
Which
Okay.
sequencing technique is that?
Okay. So, if you are asked, let me just make it clear. You are asking if there is any sequencing technique that allows calling genotypes for a 4X population for a
Yes.
tetraploid.
Yes, yes.
Okay, so I'm just seeing here that Jikin already answered this question. Technically, any sequencing technique will work for calling genotypes if you have enough depth per variant or per single nucleotide, right? So I agree with Jikin, and that's pretty much what we have been seeing here. GBS, for example, we have lots of data sets from generated through GBS. And the main issue we have for estimating genotypes is when you don't have this high read depth. So if you have a very shallow sequence depth, you don't have enough information to distinguish between the correct genotypes and the error. So you don't have a good noise to signal ratio. That's essentially what we per our experience here. So I don't know if any of you want to comment on that.
Yes, I was about to complement the answer, and we covered that actually yesterday and some other sessions in the past, like softwares that allow you to call SNPs from sequencing data and also call genotypes. So you could use softwares like JTK or Freebase to call the SNPs, and Updog, Polyred to determine the dosers, like based on the counts that you determine with JTK and Freebase. So there are recorded videos if you want to access the Polyploids website.
Great.
All right, thank you.
Thank you, Chris. Oh, and there is another follow-up question on that. So in your experience, what is the sufficient read depth? Would you like to comment on that, Chris?
Oh, it depends of the species and the quality of your data. So I don't have like a number for that. But here, like for roses or tetraploid roses, 50x, it's a good number for us. But again, it depends of many parameters like your species, your genome, your sequencing quality, but yes, it depends.
Yeah, just a comment complementing that. I think there's a study a few years ago from Jeff Endelman's group, and they simulated a lot of different read depth for tetraploids, and the sweet number was 64 or 60. I don't remember, but it's close to that, right, Chris? So yeah,
Yes, close to that.
a reasonable.
I remember that like for diploids, a good number is 6 for outcrossing. But if you go to tetraploid, yeah, it needs to be around 60 or 50.
Great.
Oh, just sorry, just the last question regarding that. So as Maria said before, for example, if I don't have that read depth, is it, I don't know, do you recommend to you diploidize the, like not calling tetraploid dosages, but rather diploidize everything to have the genetic maps?
Yes. So you're asking whether, so if you have a lower read depth, you could diploidize the data and call like 3 genotypic classes instead of 5. Is that your question?
Yes.
Okay. So yeah, it's a common approach that people have been doing in a lot of different studies, QTL mapping, linkage analysis, but it's something you could do, but you need to be aware that when you do that, you are essentially collapsing all the heterozygous classes. So if you have like a range from 0 to 4, you would have all the genotypes from 1 to 3. They are heterozygous with different levels, and you are gonna collapse them in one class if you do that. So that, in other words, you'll be losing some part of the information on your population genotype data. But if you don't have a way to increase the read depth or another way to call the genotypes, that's a good approach to go. Yeah.
Yeah,
It-
but
go ahead.
sorry. Okay. But give a try to updog software to call the dosers, even if you have low def, because it's a really nice software and it can fix like some characteristics of that we usually find in low def, like dispersion of the data, outliers, and things like that. So give a try to call the dosers using the software, and then if it doesn't work, if the def is really low, then you try it as a deployed.
Okay, thank you. But like I have been trying Updog. The thing is that I have crazy distributions in many of my markers. So I'm not sure if using that as a second alternative. Or
I see. Did you build your maps already? It's a mapping population?
yes, it is a mapping population. I have like 4 big parental families.
And the maps look good or no?
No, definitely
No.
not.
Yeah.
Yeah.
Because yes. So yeah, maybe trying deployed is another alternative.
Okay, thank you.
That's great. Another question. So Ranjana is asking if 30x is good enough. If this regards a tetraploid population, it might be a good read that you would be able to call genotypes using a good software for that. I don't know if you have any opinions on that, Chris.
She said 30x.
30x, yeah.
Yes, I would try do the same thing that Paula did, like try to call with updog and see how the maps will be. Because like usually we use the map to validate the call. So if the map makes sense, the recombination fraction makes sense, it means that the markers were called correctly. But if you can't build a map, then yes, maybe you need to do the same thing that Paula is doing.
Yeah, good. So the rule of thumb would be trying to estimate the genotypes considering the correct ploid level, especially if you have a mapping population where you can build you can perform linkage analysis. So you have a proof on the genetic behavior, right?
So yeah, it's what we do with reads to map that I discussed last week. Like the pipeline will build completely until linkage map to kind of validate. So we select the bioinformatic pipeline according to the quality of the map. But if you want to do like test just a few softwares, then you can just do this by hand without using the workflows. You use like updog to try to call your genotypes and build the maps with that. And if the maps are not good, then you call as deployed.
Yeah. Oh, that's great. We are having a lot of questions and lots of discussion here. So I don't care. We are late on the presentations. I can shorten my presentation if it's okay for you all. But we have more questions. So if you allow me, we could go through it. What do you think of using IBD from biparental populations and then use GWAS poly for QTL discovery? I don't know if that question was for Maria, because do you have any comments on that, Maria? The question is using IBD from biparental populations and then use GWAS for QTL discovery. So this IBD from biparental population would be to control the genetic background, would be, I don't know if I understood.
Yes, I'm not sure if that's the purpose of using the IBD from biparental populations, but it's populations, it's a plural. You have several biparental populations that is a diversity panel, or you can analyze are they sharing a parent? If they have any sharing parent, then QTL discovery using the QTL makes sense to me. But if these are just random biparental populations, like a diversity panel, like JK was mentioning, this is for GWAS poly.
Yeah. I agree with you. And there was a last question, as I see here. Can QTL and the GWAS poly be adapted to allo polyploids? Any suggestions for allos? Jikin said treat allos as diploid and use
Yeah.
diploid software. But
Yeah,
poly
allo polyploids are treated as diploids. You just use diploid software.
Great. All right. I think we answered all the questions, and I jump to the QTL poly session right now. So first, let me share those the code that we use for this session. As I mentioned before, this is supposed to be a short session, so feel free to ask any questions. Stop me and open your mic at any time, or drop a question here in the chat. So I just dropped the 2 links in the chat. The first one is a GitHub repository with the code and the data that we'll be using for this session. And the second one has some information about QTL Poly, which is this package we will be working with. And let me just open the first link here, and I will share my screen. Okay. Can you see my screen?
Yes.
All right. So here is the first link I just shared in the chat. It has those 2 files here. The first one has the phenotypic data we will be using for this analysis. And the second one has the R code. I will just open it here so you can see it has all the code we'll be using today. So I recommend you to download it. You can do this by clicking at this button here, download raw file. Please if you want to follow along with this session, please download both files because we will need both of them. You can just use the same button here to download the phenotypic data information. And that's it. So let me just clean the work directory here. Okay. So is the font size good?
It looks good.
Okay. So on the left side of the screen here, you'll be seeing the code that I just showed you in this GitHub repository. And I will follow along with the code. I jump some sessions, but it's okay. You have the code, and these sessions have other ways to be run. So, for example, we have 2 ways to find QTL in biparental populations. One is the manual QTL detection, and the second one is the automatic QTL detection. I will skip the manual one since the automatic performs basically essentially the same steps in a faster way. So as we have last time, it will be okay to do that. So the first thing you need to do for this analysis is you need to have 2 sources of data. One is the linkage map. Here we will be using the linkage map that was constructed with map-poly. And the second source of information is the phenotypic data, which is the same file I just shown you in the GitHub repository. Once you have your R environment set up, you can install both packages, QTL Poly and Map Poly. I will skip those 2 commands here since I already have those packages installed in my machine. And if you wanna use the latest QTL Poly development version, you can use this line of code here. So you need to install DevTools and use DevTools to install the package directly from my GitHub. Okay. So I load both packages first, and then we'll use this maps for X, which is an object that is loaded to the memory once you load map-poly up. So you'll be able to see here that the object is already loaded in the R environment. And I will use the calc genoprob to calculate all the genotypes probabilities from all individuals in this population. Again, it's a tetraploid potato population we are using. It's the example data set that we have within both packages. And map poly, we use this function calc genoprob to, based on the map, calculate all the genotype probabilities for all individuals in all map positions. Okay, so this should run quickly. At the end, we have this object here, genoprob for X. And the second step is reading the phenotypic data. Here I use the same file I shared with you in the GitHub repository. But if you were running this for your own population, I just put it here because it's something people ask us often, how to organize this data and how to read it. So let me just show you how the file looks again. So we have essentially, it can be a text file or a CSV file, a spreadsheet. It depends on the type of data, on the way you store your data. But you need to have individual names here. We have the first column with the individual names, and all the other information from the second column on should be the phenotypic information. Here we have 3 traits, so that's why we have 4 values in from the first row till the end of this file. So we have the individuals' names and all the information for the 3 phenotypes. Once you read it, it should show something like this. Let me show you the structure of the data here. So it should be a data frame with the number of observations being equivalent to the number of individuals, and the number of columns being the number of traits you have in your file. A few things that you should make sure is you have all the individuals' names need to match exactly the names of the individuals in the map. And you need to load this information as row names. So you see that if I load this, after I load this data set here, you don't see any individual names here. But if I ask row names for this object, you see all the individual names. So they need to match with the map. Here I just put 3 lines of code so you can check the individual names from the map, the individual names from the phenotypes, and another line of code with a check. So just make sure all the names are the same and all the names are contained in the other object, like all the phenotypes are contained in the map. And so the first function we from QTL Poly will be using to load the data is the read data function. You need to inform the ploid level of this population. What is the name of the object with the genotype probabilities, and what is the name of the object with the phenotype information. We have another argument step, which is essentially to inform the software at which step they need to calculate the probabilities on. And once you have all this data, you can run the function, and you see short information on the console, informing deployed level, the number of individuals, which is 156, the number of linkage groups, 12, and the step size, the map size, and the number of phenotypes, which is 3. Okay. So starting the QTL analysis itself, the first thing we need to do is determine the scored based thresholds for the forward search and the backward elimination. So usually we have a more relaxed threshold for adding QTL to the model and a more stringent one for removing QTLs, okay? So we usually do this on a data set based resampling method. We essentially simulate QTLs using your genotypic data, and we construct a distribution of the null hypothesis model. And then based on these thresholds, we can determine those thresholds. We usually recommend 20% and 5% for forward addition or forward search and backward elimination. But this varies between data set to data set, between ploids and species. So it's good if you have computational power and time enough to run this in your own data set. I just left all the functions you need to perform that in your own data set here. We usually recommend running 1,000 simulations, but here I will just run 10 for the so you can see the software running and how we get this data and use this data in the further steps. But if you were running it in your own data set, please run with a higher number of simulations, okay? So let it, so okay, the simulate QTL function, it's really fast. It runs very quickly. And then you need to run the null model based on the data set that the previous function generated. If you have QTL Poly installed from the development version, from the GitHub repository, you can use the null model 2 version of this function, which is faster. And I use this because we have limited time here. So let's just wait a little bit so it will run for the 10 simulations. While it runs, if you have any questions, please let me know. Open up your mic, or I don't know if we have questions on the chat. I cannot see any here. So another thing, if you want to run this in a real scenario, you will be running like 1,000 simulations. Please use the newer version because it's way much faster than the previous one. Just so you know, for the sweet potato data sets that we have been running analysis here on, we usually spend like 1 week just for this step. And with the new version, you can run this in less than 1 day using a high-performing computational cluster. Okay, so it just ran. We have a new object with called score null with all the information we need. So with this following line, we can get the distribution of all the minimum p-values for all the simulations we just ran. And we can have this, we sort these values and we get the quantiles back. As I mentioned before, we usually use 20% and 5% for forward and backward steps. Those values are as follows: 0.005 and 0.0003. I just save them as sig forward and sig backward. And then you can run, as I mentioned before, you have 2 ways to run the analysis, the manual one and the automatic. I will skip manual and I would jump into the automatic way. The results will be the same, but the automatic is faster. And also we have a new function, remin2, which performs way much faster than the previous one. So if you are running in a real data set, especially with a higher number of individuals, please use the new version, which will be way much faster. Okay. You have a lot of arguments here. The first one, you need to indicate where the data set is, which we saved as that. The second one is the phenotypic column. Here you can select to run in only one or a few traits or all the traits. If you want to run in all the traits, you can just erase this part and it will run in all phenotypes that are available in the data set. Then you have the window size. The default is 15 centimorgans. You have the parameters for the significance thresholds for the forward search and the backward elimination. Also, you have the interval, the support interval, what would be the factor to use to determine the support interval for each QTL. And you also have these N clusters. If you have a powerful machine, you can use a higher number of clusters and the analysis will run faster. In this case, I run with 12 cores, one for each chromosome. And I selected only the first trait, so the analysis runs faster. So let's see how it goes. This step usually for a hexaploid population, we use it to take like many hours, like 8 hours, 10 hours. It depends on the number of QTL. As you add more QTL in the model, the analysis goes slower. And if you have a higher number of individuals, it goes even slower. So again, if you were running this in a real scenario, please use the second version of this function, remin2, okay, which is available in the development version. The software let us know how the analysis is going on. It is informing us that one QTL was found on the linkage group number 5 at 26 centimorgan, and another one on linkage group 7 at 60 centimorgans. All the steps that you were seeing here, like the search, the refining, and the profiling, all those steps, you need to run one function for each one of them in the manual search. But for the automatic function, which is the remin one, you don't need to worry about it. The function will run everything for you. So, okay, it's finished. You can use this print on the object that you just saved, and it will tell you the QTL information, the linkage group number, the position, what is the mark number according to your data set, the marker names, the score, the score statistics, and the p-values for each one of the identified QTLs. If you want to get lower and upper boundaries or support intervals for the QTLs, you can just run the same print function with this argument sint, lower or upper, depending on what you want, and it will return you the lower and the upper boundaries for each one of the QTL. You can also use the same data set to plot the profile of the QTL. Here, I just show you for this first trait. Oops. Let me generate it again. So we have the 12 chromosomes, the profile, and the QTL peaks and the location on the chromosomes. And you can also see the support interval for each one of the QTLs here with the plot sint function. If you're done with the analysis, you can just fit the final QTL model where you have all the genetic variance and the residual variance estimated. You can have the heritabilities calculated. Here, let me show. Okay, so we have for these two QTLs, multiple QTL model. It fits this multiple QTL model and return you the information regarding each QTL, the intercept of the model, the genetic variances, the residual variances, and the heritabilities for each one of the QTL and the total heritability. Okay. You can also use this QTL effects function to calculate the effects for each one of the alleles. Here, I will show you the second QTL only. So we have both parents. A, B, C, D represent the haplotypes at the QTL position for the first parent, Atlantic. And E, F, G, and H are the haplotypes at the QTL position for the second parent. And you can see how the effects are associated to each one of the haplotypes in this population.
Gabriel,
Yes?
may I ask one quick question?
Yes, please, go ahead.
Sorry to bother you, but I have a question about your QTL research. So would you take a look at trait 1 F107? So in this case, you found two different QTL on chromosome 5 and 7, but sometimes I also saw why I'm using the QTL poly.
Mhm.
Can you see the number 3 NA not available for like with a certain number? So do you know the reason why those kinds of things take place?
Mm,
Yeah. That is what I'm looking.
so if you can clarify, I don't know if I got your question correctly. Can you say it again, please?
Oh, so
Mhm.
my question is, I suppose that the QTL poly found two QTL on chromosome 5 and 7, but when you see the third row, you also start to see some understandable numbers, like an intercept 72.9276 or something like this. So I also saw similar cases before why I'm using the QTL poly. So do you know the reason why this third row took place?
Oh, do you mean this row here, the third
Yeah,
one?
that row, yeah.
Oh,
Exactly.
okay. So it's meant to be the information because it's a multiple QTL model, right? So you have only one model with two QTL effects. So you have one variance component for each one of the QTLs that are here,
I
and
see.
the first and the second row, and you have the residual here. Okay.
Oh, is that the residual? I see. Oh, okay,
Yeah,
got it.
yeah. Because it's a multiple QTL. It's not you are not feeding the model once for each QTL. At the end, when you are done with the analysis, you re-estimate all the effects with all the QTLs in one single model. So that's
Oh,
why we have only one intercept, two genotypic variance components, and one residual variance component.
I understand. Thank you so much. Yep,
You are
got it.
welcome. You're welcome. You're welcome. Thanks for your question. Okay. And last of all, I'm sorry, Peter. I'm taking a little bit of your time, but just to finish up here,
No
you
problem at
can
all.
also use the breeding values function to calculate the breeding values according to the QTL you just found in your population. Okay. So it will generate a vector with the adjusted values. Here you can plot and see how is the distribution looks. And you can also get like a higher, the individual name that is the higher performance in your population. For example, here is the individual number 146. I will skip the fixed effect model here, but you have the code. If you want to run this, you can perform the permutations and run the fixed effect model for comparison purposes. And the last section here, you can save all the objects and use in the ViewPoly software, which Chris will be talking about in another session. So that's it. I'm sorry again, Peter, for taking your a little bit of the time of your session. I will stop sharing my screen. And
You have
I
another
guess,
question, Gabriel?
yeah, I guess we don't have time for questions, but feel free to drop questions in the chat. I'll be happy to help. And Peter, so Peter,
Hang on, can you hear me?
yes, we can hear you. We'll have you presenting the next session now. Are
Maybe,
you
but yeah,
are you
maybe
good to start your session already?
sure. I mean, if I'm pretty relaxed here, so I mean, I can. I think there was one question also from Jay about how to decide the significance thresholds, which you already showed. But maybe you
Okay.
could say something about the choice of those alphas, so the 0.2 and 0.05 maybe,
Yeah, sure.
perhaps. I don't know
So
if that's what the question was.
yeah. So I don't know if that's the question, Jay. I just repeated how to decide significance threshold for both forward and backward while using Reming mod. So for this, for the random effect model that we are using here for mapping QTL, we usually recommend using the 20 and 5% thresholds. but those numbers will depend on your population, on the ploidy level, in your data set. So that's why we recommend using the resampling method. So you can account using simulations and resamplings on your population. You can construct a node distribution for the null hypothesis where you don't have any QTL in your population, and use this more relaxed threshold, which is the 20%, for the forward search. So you allow more QTL to enter into your model. After you have all the QTLs in your model, you have a more stringent threshold, which we recommend using 5% for removing those QTL. But again, the p-values that we will use for that will depend on your data and this node distribution that we will construct using the resampling method. So they are just a general recommendation, the 20 and 5%, but you can change it according to your population again. It is based on simulation and how we have been searching for QTL in both tetraploid, hexaploid, and octaploid populations that we have been working here. So I don't know if that answers your question. If you have any other questions, just drop a message here. Peter, so welcome and thanks for your time. Again, sorry for taking a part of the time of your session. And if you are comfortable and you are ready to start your presentation, feel free to take the floor. Mm-hmm.
Thanks, Gabriel. Last week, I ran something similar, which is just to first of all maybe open it up to people if they have questions, just to throw any questions at me. Also, while I'm talking, feel free to interrupt. I presented last week a couple of slides. I'm not going to do a live session just like Gabriel did. For that, you'll have to wait until January at the SCRA, the next workshop, where I will be presenting also a sort of a live workshop where we go through some things together. But today I just want to refresh a bit what PolyQTLR is and what the package can do. Again, the background, the package was developed originally as part of sort of a domestic Polyploids project we had, and we had a number of them. But the PolyQTLR really took shape in what we would call Polyploids 2. And then also it further was developed and extended during our involvement with the SCRA initiative over the past number of years. And also just to remind that, I mean, a lot of the things, the basic tools, the basic functionality of the packages, they've already been presented in previous sessions. So there's a bit of repetitivity or repeatability or repetitiveness in what I'm going to be saying. But just to remind people that there's a lot of those presentations from previous years already on the SCRI website, polyploids.org. So do have a look there if you're interested again. In terms of sort of taking a sort of a broad look about what we do in QTL mapping, we do something relatively similar to the other guys, our guys and girls in a sense, in that we estimate these IBD probabilities. We use them in a model. After which we then, once we have detected QTLs, we fit them as cofactors to see if we can detect other QTLs, so building a more multi-QTL model. Of course, we have to find whether QTL exists or not, or whether it's significant enough to be considered a QTL in the definition of a QTL here. We estimate significance thresholds using permutation testing. And afterwards, once you have done those steps, you end up with a sort of, a multi-QTL model, which you can use for further purposes for breeding. In terms of these steps to go through them, there was already talk about sort of at the heart of all these methods, really using populations with the known relationships. We estimate these posterior genotype probabilities, what we often refer to simply as IBD probabilities, as a more complete representation of your marker data. So as Maria mentioned, you move away from using your marker genotypes to your IBD genotypes. And they are, of course, extremely useful for association analysis. And I think it's maybe worthwhile to spend a little bit of time as to explaining how we estimate them. And so the original approach to do this that we have incorporated in PolyQTL or was proposed by Xiaozi Zheng, who also worked on PolyOrigin. But so PolyQTL or uses the same hidden Markov model that was originally built into TetraOrigin, which is the sort of the forerunner of PolyOrigin. And the idea is that when you have a single individual, in this case, this little plant, we don't really know at the outset how that plant was formed. So what happened in the parental meiosis. So basically, PolyQTL or this hidden Markov model goes through all the possible pairing combinations within the parents and comes up with, well, it builds that into a Markov chain to figure out what's the most likely pairing scenario within the parents that gave rise to this individual. And of course, well, we can either consider in an autopolyploid the formation of bivalents. So this is where the chromosome copies pair in pairs, or we could also consider the possibility of a multivalent. If we consider a bivalent, which is just a simpler thing to visualize here in the slide, but then we can go through each of these possible combinations. So for instance, in this combination that's shown here, if we have these numbers 1, 2, 3, 4, where 1 and 2 are pairing and 3 and 4 are pairing, well, then we know immediately that the possibilities of alleles carried on haplotype 1 and 2 from the mother is not a possible combination here because they're pairing, and each bivalent will give rise to, will send on into a gamete one of these two chromosome copies. So just basically by doing all the sort of the combinatorics, you come up with various different possible matrices. So these emission matrices, and so they're built within this Markov chain for each possible pairing configuration. And as I said, in PolyQTL, we allow the user to decide whether bivalence is the best way to model it, or to also allow the possibility that that multivalence, in which case you have more of these genotype classes available. And then that's of course done at each position. And we build into these emission matrices then the probabilities of observing the dosages, so the genotype calls that we see in that individual, allowing for the fact that we might also have genotyping errors. So we also model an error possibility, which allows us also to accept errors in our marker data. On the other hand, we have a genetic linkage map. So there was questions about whether you use, if you have only a physical matching rate. That question came up. Well, the reason for the necessity of having a genetic map is that we need to know something about the probabilities of moving from what's called one of these states to another state. And that's handled in this Markov chain using these transition matrices. So a transition matrix allows you to move from a particular state, a particular parental configuration of alleles, to another one through the process of recombination. And we only know something about those probabilities of moving from one state to another if we know something about the genetic distance rather than the physical distance. So we need to have these recombination frequencies, which are these probabilities of moving from one of these states to another. And of course, if your genotype data is telling you that you have to move through a couple of states, well, that's less probable than moving maybe just from one state to a nearby state, because these probabilities multiply, and it becomes increasingly less likely of moving through multiple states. And so ultimately, there's some sort of an algorithm which integrates this information called the forward-backward algorithm. So that's essentially multiplying these matrices together in a particular way and summing probabilities. The net result for the end user, at least what the output is or one of the main outputs is these IVD probabilities, which can be visualized like these. So these are, in this case, the haplotype probabilities. So the probabilities of inheritance of particular parental alleles. Now, PolyQTL uses a slightly different color scheme, where here the blue is a single copy, and regions which show red would be carrying a double copy of that particular parental allele. So once we have those estimated, we then use them for trait mapping. And essentially the model for that association model is basically a weighted regression using these IVD genotype probabilities. And that's performed genome-wide, a sort of a QTL scan. So again, quite similar to what's done in the alternative packages. Up to now, we have only used an additive model for the QTL detection step. So I'm going to be showing about a small extension to also doing an initial scan for non-additive effects, and that will be presented in January as well. As I said, we use permutation tests for setting our thresholds. It's fairly fast, I would say, the implementation. I don't have anything to show you here, but all of these steps are quite fast. And there are particular tricks that you can use. For instance, if you have a very high density data set, we often, what we do is we spline our probabilities at a regular grid of positions. So we reduce from maybe 1,000 positions on a chromosome down to 100 for this initial QTL scan step, because you must understand polyQTL is developed for biparental populations. So there will be a limited number of recombinations in such a population anyway. So having many up to 1,000 or more loci on a chromosome doesn't make much sense. You don't have that amount of genetic resolution. So we do things like essentially interpolating our probabilities at a regular spaced grid, and that allows us then to speed up a lot of these steps incredibly. We also recommend the use of blues, particularly if you have a data set with, for instance, blocking factors, or you're including genetic cofactors. A lot of these steps can then become a lot slower. So if you use blues, and that's again described in the vignettes, and I'll be showing it again in January, that can speed up this step a lot. And afterwards, once we have detected QTL positions, we then explore those QTL peaks. For instance, looking at the most likely QTL segregation type, we do various visualizations of homolog effects, and ultimately a user can use the package to also automatically build a QTL model. And that was again something that I showed, I think maybe already 2 years ago, the automatic cofactor function within the package. Just to give a couple of examples, we had a study a number of years ago in rows, and this is the kind of the output that you get, where you have, for instance, various QTL detected for, in this case, the number of prickles on the stem of rows. We found fairly large effects QTL on chromosome 3, and we can see particular effects, and the effect size in this particular visualization shows that on homolog 3 from the mother, there was an allele which contributed to an excess of prickles in the offspring. To say, after you've found some QTLs, I mentioned these cofactors, that can either be done automatically or you can do it manually. You can play around a little bit with adding different cofactors and seeing what effects that has on other QTL peaks. So this can help to uncover additional QTLs, as I said, but also you can see in how different QTLs interact with each other. Or you may be interested, if you have quite a large peak on a chromosome and you're not quite sure, is it a single or multiple QTL? Well, again, it's quite easy to fit one peak and see what happens to the second peak. So just to give a couple of examples of this from a previous study we had in chrysanthemum. Now there's two different lines here. One is HMM, which is the hidden Markov model based IBDs. The other, which we used to call the quick and dirty method, Q&D, is now called the heuristic method. We've become a bit gentrified. But in any case, it's a much faster way of sort of estimating IBDs. So, well, first thing is that there wasn't a huge difference in the outcomes, whether you use the hidden Markov model or not. For a hexaploid, especially if you allow 4 multiplets in a hexaploid, the IBD step can become quite slow. And that's, in that case, and we showed it through simulation. If you go up to ploidy levels of higher than 6, then then the timing for you use the heuristic method in those instances. But anyway, to get back onto the point in hand with cofactor fitting, for flower color in chrysanthemum in this population, we found 2 major QTLs. So we were able to fit them both then as cofactors in a second genome-wide scan then. What you see here is those 2 major peaks in green have disappeared, and what we picked up is this extra QTL on linkage group 9, which again, well, it was borderline significant before we had fit them as cofactors, but here it pops up. And these threshold lines that you see here are recalculated each time, so they're all recalculated based on a new model which includes cofactors. So that's something that's important to bear in mind, that we're not cheating here. This threshold is actually valid for all, in this case, for the 2 different models that are visualized. If we look at this other situation for the number of florets, what's interesting here is if you look at linkage group 3, you can see that they're in green again. There were 2 particular peaks discovered. So this could be, again, a situation where, well, you don't really know what's going on here. Is this 2 QTLs? Do I have 2 genes at both ends of this chromosome or not? So again, you can just choose one of those peaks as a cofactor, very simply add it. And in this case, yeah, you see that when we fitted the first of those peaks as a cofactor, the second one, although it drops below the significance threshold, it still remains, I think, quite an interesting locus. So, I mean, it is being boosted to an extent by the other QTL, but this is clearly an example of where there are 2 QTLs on 1 chromosome for this trait. Just to briefly mention some other functionality in the package. I mean, we were talking about QTL analysis, but you can do an awful lot more. You can look at, for instance, recombination. PolyQTL gives the user information on recombination per chromosome, which could be interesting if you want to see particular areas that are hot spots or cold spots for recombination. But what we also really recommend is to also look at the recombination counts per individual, because that often shows us individuals which have far more expected recombinations than you would expect by normal processes. We can also pull out recombinant individuals, which can be interesting as well if you want to combine different haplotypes in an individual. Application that I talked about again at one of the previous SQRI sessions was about genotype correction. Again, once you have these IBD probabilities, you essentially have a very good picture of what was inherited in each individual. And if your data set is quite noisy, you can use the fact that by modeling it with a higher error prior, you essentially smooth out your genotypes, and this can be a very powerful way to re-impute marker dosages if you have very noisy data sets. And it has been, we've shown it in the past, it can not only improve your linkage maps, but also improve power for QTL detection. And another application that I think is very interesting is the study of meiosis itself. So processes or things like preferential pairing and double reduction. Just to give an example of what we found, for instance, in chrysanthemum, which is again an interesting hexaploid species, quite an important ornamental crop, certainly here in the Netherlands. We re-examined a data set that we had produced from a previous study, so really very nice hexaploid data set. And we found a very nice picture of what went on during the meiosis in that population using PolyQTL. What we found on linkage group 1 and 5 was that in some of these cases, so what we see here, just in case you're wondering what are these little hexagons, these are the different parental homologs. So each of these boxes refers to a different linkage group. And these numbers A, B, C, D, E, F, they would be the 6 copies of linkage group 1. So that's the mother, and then the second I, J, K, and so on are the 6 copies in the father. If you see in linkage group 1 and 5, we found a very strong preferential pairing between, in these cases, 2 of those homologs. So this meant that in many more meiosis than you would expect by chance, homologs J and H were pairing and recombining with each other. And that also, we picked that up also on linkage group 5. Now the numbering J and I, that doesn't mean anything. They're just numbers. They could be, that doesn't, you know, it's just a number to code a particular parental homolog. But there was those 6. What was maybe even more interesting was what happened on another set of chromosomes. Again, only in the paternal meiosis, so only in the father, this was happening. But linkage group 3 is a very nice example of where, again, you see these preferential pairing coming out with these red bars. So this is an excess of pairing between these homologs. But here you get a configuration where they fit nicely into 3 pairs. So this is reminiscent of an allopolyploid type behavior. But chrysanthemum is one of these ornamental species that's had a lot of hybridization across species in its pedigree. So there is, again, I'm not a chrysanthemum guy as such, but I know there is still, even in the literature, controversy about whether it's an auto or an allopolyploid. And we were very strongly suggesting it was an autopolyploid because that's what the marker segregation looked like. But on closer looks, there is also some residual effect of allopolyploidy in the ancestry, and some sort of subgenome type pairing behavior going on. So that's, again, this is the sort of output that we like to look at. And another application than just QTL mapping. And I think this is also something that I'd like to finish on. Are there questions? I know I'm almost out of time, but I'm happy to take questions. And other than that, please, if you're in either in San Diego, I won't be joining myself live, but I will be online in January. So please, hopefully you'll be able to attend the updates and the workshop in January for PolyQTL or Polymapper.
Nice. Thank you very much, Peter, for your time and your presentation.
Stop sharing this.
Yeah, if anyone has questions, please send it on the chat or open up your mic. We have 2 minutes, one minute left. Okay.
Must have been either very clear or very boring, but in either way, no questions. Thanks, Gabriel.
Thank you very much, Peter. You're super clear. I think we can start the next section. Alejandro, you'll be talking about MPQTL. Alejandro, if you are ready, please, floor is yours. You can share your screen.
Yeah.
Hello.
Can you hear me well?
Yes, we can hear you well.
Okay. Let me share my screen. So you should be seeing now my slides, hopefully.
Yes, we can see it.
Let me get out all of this annoying stuff. Okay. All right. Let me put the pointer and then I'll get started. All right. So indeed, I will be talking about MPQTL. So MPQTL is a tool for multiparental analysis or more multiallelic analysis. You will see in a moment. We have some examples of some polyploids that I liked. I found this cover slide among some of my presentations. I thought it was quite pretty. But I'm not going to be talking about flowers. I'm mostly going to be talking about statistics. And I would like to start with an introduction on why we made this software, what was the concept behind it, very, very briefly. So for mapping populations, I think the very classical mapping populations are biparental mapping populations, right? You have 2 parents, they contrast on some phenotype, and then you have some population in which you map a trait. Hopefully you find a QTL or more. And of course, on the other extreme, we have association mapping studies. So the big difference between the two, I would say, is in the biparental mapping, you know the pedigree very well, so you know what frequencies for your alleles you can expect. Whereas in association mapping, so in GWAS studies, in the end, the population comes from a very complex pedigree or a natural population, basically. So the frequencies of the alleles that you get are very, very unknown. Of course, even though that can give some statistical problems, but that also means that we're evaluating in a GWAS study way more of the genetic diversity of that of the organism compared to what we get in a biparental population. So these are two extremes, right, of the types of populations that we can make. While I was researching, so these are indeed two extremes, and I think in between point between one and the other are multiparental populations. So multiparental populations are also known as connected populations, or sometimes you will hear of family mapping or pedigree-based analysis. All of it refers to the same thing, a set of biparental populations that share some common parents. So the simplest multiparental population would be just two biparental crosses that share one parent in common, and then you already have a multiparental population. For the study in which we developed MPQTL, we tested our model with something called nested association mapping, or NAM populations, in which you have one central parent and several peripheral parents. And basically, the population consists of several crosses that share the same parent. In the end, all of these, of course, also includes breeding populations, right? Breeding populations are also sort of multiparental populations, because in the end they are formed by biparental crosses that are connected by some common parents. Now, why are we interested in multiparental populations? Because on the one hand, they have more diversity than biparental populations, so they allow us to assess a broader range of alleles than in a biparental population, but they're not as messy as diversity panels. Right, the frequencies are more well known, and you can design it a bit better to represent the diversity in a more balanced way, compared to in GWAS panels, where you might have some individuals with extremely rare alleles that are very difficult to assess, and if they contribute something to a phenotype, they're hard to detect, and so on. So that's a bit the basis of this. Now, the key aspect of doing mapping in a multiparental population is the difference between, well, can be illustrated by the difference between biallelic models versus multiallelic models. And that's actually the key innovation of MPQTL, that it can use multiallelic models. So to illustrate this, let's imagine like an example population of a very heterozygous tetraploid biparental population. So in this case, we're not even looking at a multiparental population. Now we have these two parents, and they're so heterozygous that in each different chromosome, they have a different allele. So for one locus, let's say parent one has, in one locus, parent one has 4 alleles, and in the same locus, parent two has another set of 4 alleles. And we have some linked SNP markers, of course, some random markers next to it that are linked to each of these alleles. Now, in a classical biparental analysis, so using SNP markers, what you would do is group all of the individuals that have either the A or the B allele and separate them into groups. So this would look like something like this. So you have the alleles in the group A, which are these from parent 1 and these 2 from parent 2, and the alleles in group B. And you would separate your individuals and do basically a 2-group test, right? You separate individuals into groups and you see if this separation is meaningful. If you only have one allele, or if you have very few alleles, maybe the separation is meaningful. But when you have many alleles with different allelic effects, you can see that, yeah, why would this grouping give us any significant signal, right? Even if this allele has some relationship with the phenotype. Because, if these alleles all have very low effects, and if these alleles have all very high effects, in the end, by creating this artificial division into groups, we end up conflating ones with the other. And the same would happen with the second allele, and the same would happen with the third SNP, right? But if we combine them, so if we combine 3 SNP markers one after the other, if we haplotype them basically, we, instead of creating this biallelic, these two groups of markers, we create suddenly 6 groups of markers, and we can distribute each allele effect into one of these groups. There might still be some alleles that end up in the same group because by chance they have the same haplotype linked to them. But in the end, we move to a multi-allelic situation where we're closer to the reality, which is that there are 8 different effects. So we're modeling 8 different effects, or in this case, 6. Of course, the more SNPs you add, the more alleles you have. So was a very, very short, what is the purpose of MPQTL? Is to try to model these sort of situations. The key, of course, is that these only make sense when you have a lot of alleles, when you expect many alleles in your population. So in very homozygous diploids, for example, it's probably not very useful. But in very heterozygous polyploids, where there are very likely many alleles in the population, then is when these models start to become very useful. The nice thing of haplotypes is that if 2 individuals have the same haplotype, they are very likely identity by descent. So they're not just equally identity by state and that's it, but you can prove that it is very likely that they are IPV. In any case, again, this was only the theoretical introduction. Then what we did for the QTL analysis, can ignore this, was just use the unified mixed model. It's very classical, typical model. You have the genetic effect as a fixed term and the kinship structure for the kinship correction as a random term. And the only thing we did was adapt this genetic matrix so that it could use this multi-allelic modeling and adapt the calculations for the relatedness matrix using haplotypes instead of using biallelic markers. And now this has several advantages. So one is that we can estimate the haplotype effects. And as I mentioned, haplotypes are quite close to IBD. So that means that you essentially are estimating the IBD effect. You can join information across multiple biparental populations. And of course, you can locate and characterize QTLs, which is the whole point of QTL models. There's one example that I really like to illustrate why any of this is better in situations where there is high diversity. And it's this figure. So this is from the paper of MPQTL. You'll probably find it better explained here, but in the paper, I mean. But let me give you a short summary of what it means. So basically, we're saying at a simulated population where there is one QTL in this position, and here, and here. This is just the same population, but we're doing the QTL study either using SNP markers, true IBD markers, which we know because it's a simulated population, or using haplotypes. Haplotypes that are just the concatenation of subsequent SNPs. And what you can see is that when you use biallelic markers, again, this is a high diversity scenario, you get this very messy set of significances. You have many markers that are nearby the QTL position that are not significant at all. You have markers that are far away from the position that are significant. And in the end, you end up having these QTL intervals that are pretty large. And in this case, the most significant marker is here, but it could very well be that the most significant marker, the peak marker, is far away from your true QTL position. In the case of both IBD and haplotype markers, you see that there's more this sort of monotonic increase of significance as you get closer to the QTL position, and then decrease as you get away from the QTL position in both cases. Yeah, so that's it. And you would expect this difference when you use multi-allelic or biallelic models in these situations where you have many allelic effects in your population. And that's what MPQTL helps you to do. So try to model these sort of things. So that's it. That's the introduction, theoretical introduction that I wanted to give. So I don't know if anybody has questions about this. I can also talk a bit about the software itself, but maybe it's better to get anything out of the way now.
Thank you very much, Alejandro.
Okay. Well, So I also have some slides about how the software works and what functionalities it has. If you want, I can talk a bit about this.
Yeah, that's nice.
Okay.
We have, yeah, Tim just said he would like to see those.
Okay, good.
But before you go, can I ask a question?
Of course.
So when you go into multi-allelic markers,
yeah,
and the population size of each of those classes is going to be affected. When you have biallelic markers, you've got basically splitting your population in two pieces.
Yeah.
If you have multi-allelic, you've got to split it into more pieces.
Yeah.
What are the issues of power of detection of QTL if we go multi-allelic rather than biallelic?
Yeah. So that's a very good point. To be fair, we didn't calculate exactly what happens in terms of power for each individual allele, but rather as a whole. So as a whole, I can tell you in terms of power, you detect more QTLs and you detect them more accurately in the multi-allelic scenario. And that's what we showed in the paper. For individual alleles, as I say, we didn't do any, let's say, formal testing, but what we got from our research was, in the end, it depends on how many individuals have that particular allele. So I like to think of each allele being estimated or having the power of how many individuals contain that allele, and also how many copies. So some alleles, some haplotypes, essentially, will be less frequent in your population. And for those alleles, you will have less power than for the ones that have more individuals containing those alleles. This happens, or this is very clear when you have a multi-parental population. It's very common that you have 2 or 3 crosses that are huge and other crosses that are very small, right? Have many individuals. If those smaller crosses have less unique alleles or alleles that are only present in one of those crosses, then those alleles you will estimate their effects with much less power than with the very common alleles. And that's also what we saw when in some of the simulations. I don't think I get into this in the paper, but in some of the simulations that happens. And then you see that one of the QTLs that we were simulating was much harder to detect because most of the important effects were in populations that were relatively small. So indeed, that's a bit what happens. You essentially need to see what is the frequency of each multi-allelic marker of each haplotype.
So you would say that the method would underestimate the relative importance of rare alleles in the population. Those tend to escape us, right?
Well, I mean, yeah, that's well known, right? That's the same thing that happens in the GWAS panel. It's just that in a diversity panel,
yeah,
you just assume you're not going to find it, or you're not
but
going to.
you would exacerbate that with multi-allelic markers versus biallelics.
Let me think. Would you exacerbate it? I don't know. I don't know if you would make it worse necessarily. If anything, it would be easier to see what is happening.
So don't you think that if we had biallelic markers, we would somehow track that segment as being important in any case? Well, with multi-allelics, we may not, or.
Well, I think
I need to think about this a bit more too, because it's an interesting approach. Mm-hmm.
Yeah, but I think with biallelic markers, you depend on the fact that, so let's say we have one very rare allele. If there is one SNP marker that tags uniquely that allele, that SNP marker will be very rare. And then you will not have any effect. You might not have any SNP marker that tags uniquely that allele. And then you're confounding the rarity of the allele with the fact that you're mixing apples and pears, as they say in the Netherlands. You're mixing effects of one very rare allele and one less rare allele, but that has nothing to do with the first. So yeah, I don't know. To be fair, we didn't test this super thoroughly, but what we see clearly is that at least in multi-parental populations, the multi-allelic models are more powerful.
Okay. Thank you.
Yeah. Thank you for the question. Any other questions? Otherwise, I can just give you a few pointers on the software. Take that silent.
Yeah, if you can do that. Let me see. There's another comment here.
I don't see the comments because I don't
Yeah,
know.
so it's Tim. He's saying that anecdotally, last time tracked by allele QTL in microhaplotypes, the alternative allele was only present in a single haplotypes. So yeah.
I don't know what that means. Last time tracked by allelic, I tried the allelic microhaplotypes.
Hi, sorry, it was just emphasizing what you described there. That as it's very anecdotal because it's a single case, but I was looking at a colleague had picked up on a QTL for a biallelic marker, and I looked where it lined up against a microhaplotype assembly, and like you say, the biallelic marker that had been picked up, the alternative allele at that locus that it was picking up was present only in one of the microhaplotypes of several. So it's pretty much what you just described.
I see.
Although very, very anecdotal because it was just a single analysis.
I see. Thanks. Yeah, now I understand. Good. Wow. So thank you. But I think I will mention something about the software. So you can find it on GitHub. We've been trying to put it in CRAN, but there's some part of the functions that they really don't like how we coded them, even we did our best. So we're trying to see how we adapt it to get into CRAN. But in the meantime, it's on GitHub. There's instruction there on how to install it. You can also find the vignette linked in the GitHub, which this is the address of the vignette. The examples I will show are also covered in the vignette a bit more in depth. So you can also look at it in your own time. I don't want to spend a super long time talking about all the functions, so I'll just quickly go over the types of data that you can use. So these are the data that you want to have in order to do an analysis with MPQTL. You can use SNP data, so biallelic data, either in the form of so these are typical allele dosage table, markers on the rows, individuals on the columns, and as of ploidy, it can use any ploidy, odd or even. I don't think it will matter for this analysis. And yeah, any ploidy you can use either discrete genotypes, or if for some reason you have continuous genotypes, it will also work with continuous genotypes. So in that sense, it will not complain. You can also use, and that's the interesting part, haplotype data. The way that you code haplotype data in this software is basically also a table, but then we have a column for each of the parent chromosomes. So for example, in this case, it's the individual called A1P1, and is the first chromosome of this individual, second, third, and fourth. It's better if you use this coding, so the underscript, underscore, and then the number of the chromosome, mostly because some of the naming functions internally, they expect this naming, and then the names can be propagated throughout the output a bit better. What this contains here is just the name of the haplotype. So here are numbers, but they could be names as well. So haplotype A or B or C or whatever you want. Here, of course, this is from the simulated data, so the data is phased. So all of these markers are on the same chromosome. In terms of the analysis, this doesn't matter. Yeah, that's what I just said. So the phase does not matter. So I could put the 28 here and the 69 here and the other way around, and it wouldn't matter for the software. So you don't need to have phased haplotype data. You just need to have some type of multi-allelic data. Then besides this, you of course need some kind of genetic map. You can either use centimorgan positions or base pair positions. Yeah, the column names must be marker, chromosome, and position, but that's also described. Again, you can use positions either as centimorgans or base pairs. If you do use base pairs, then you need to change some parameter in the kinship calculation, which I will explain in a moment. Yeah, and you can include unmapped markers by adding a chromosome 0, but you still need to give those markers some position, because yeah, otherwise it will complain. All right, the last thing, of course, is the phenotype matrix. So you can use either a vector or a phenotype matrix, and where each column is a phenotype and each row an individual. So something like this. For example, this is the example data set that is in the software package. So there is an example data set with a package right away. So you can also just test it or look at how the data is shaped by looking at that data set. I always like to do that. Okay, but maybe more interesting than talking about the shape of the data is talking about what is the functionality of the software. The main function is called map QTL, which maps QTLs, not very imaginative. And it can basically run 4 different models, 4 different versions of this mixed model, either without the mixed aspect. So these are 2 without the mixed aspect, so they're linear models, either with no structure correction or with what is known the Q structure correction, which I'll mention in a second, or the mixed model with K structure correction, or if you use both this Q term and the K term, then it's called Q plus K. Common notation, but I don't know how accurate it is. In any case, all of these models can be run either in biallelic mode or in multi-allelic mode. It will run regardless, and it's very easy to change from one to the other. So let's say we're trying to run a linear model with no kind of structure correction. We just give phenotypes, genotypes, diploidy, and the map. If the genotypes are some SNP dosage, it will tell you SNP dosages have been detected. More accurately, it should say biallelic markers, but it says SNP dosages. If you give a bigger table, basically, so a table where the number of columns is the number of individuals times diploidy, it will assume that you have given haplotypes, and then it will do the models in a haplotype form. So in that sense, simple, very easy peasy. The results that you will get is something like this. So with the list, each item in the list is for one of the phenotypes, and you get beta, which is a list of effects for each marker, an F-test score, p-value score, standard error for the beta estimates. Each item in this beta will be something like this. So this is the intercept, and this is the marker effect. If you have haplotypes, there will be multiple marker effects, each with the name of each of the haplotypes, and one intercept as well, I think. And it's all effect of all haplotypes except one, I believe, because it's like a standardization process. But yeah, so this is the basic output. Now for the kinship corrections, the different types of kinship corrections, so either Q, K, or Q plus K. The Q, if you're not familiar, is just like a population identity type of correction. So say that you know that in your population that these individuals belong to this subgroup and these individuals belong to this subgroup. You could use such information to create a very simple structure correction that is much more efficient than using the kinship correction with a mixed model. So for example, I have this population vector, which defines the first 3 individuals belong to population A1, and the second 3 to A2, so on. So you can use that to do the Q correction. Doesn't work super well, but you can do that. You can see the kind of matrix that is created with this function that is not exported, because I wouldn't recommend you to use it. You can use it directly from the map QTL function. I will show you that in a second. The other type of structure correction is the kinship matrix, so the mixed model version, and we base this on the realized relationship matrix described in the GWAS Poly paper. Is a sort of relatedness. So if you look at the K matrix, which you can, it's not really a distance measure, but it's very close to it, and it's fast to compute, and it's useful for the kinship correction. But just so you know, if you look at the values, they're a bit strange from what you normally get with other kinship measures. And again, can be obtained with SNPs or haplotypes. You can use this function calc k. Importantly, you need to give the genetic data transposed. So instead of the markers on the rows, you get the markers on the columns. This is because distance matrices in R are all computed like that. So the observations are on the columns and the individuals on the rows. So I wanted to keep that. And as you can see, you can either do it with a SNP dosage matrix or with a haplotype dosage matrix. And you just need to add haplotypes equals true on the ploidy, and that will be it. Okay. Before moving on, maybe you want to see whether the dosage and haplotype matrices, they give the same result. And this is the genetic structure image that you can get. So this is a principal coordinate plot from the dosage matrix and the haplotype matrix. As you can see, they're almost identical. So just to show you that it doesn't matter whether you use one or the other for this kinship calculation. An interesting thing is that you can use these to create a Q correction. So instead of using some vector that defines this is this population, this is this subpopulation, you can use a decomposition of the kinship matrix to do this Q matrix. This was proposed some time ago, and it works pretty well. So if you don't have any population information, but you want to use the Q correction, you can tell that to map-QTL. So you can use map-QTL, and if you put Q equals true, then it will use this QPCO method to calculate the Q correction. If you give a population vector, it will use the population vector. If you put K equals true, then it will use the kinship matrix method. If you set both to true, then it will do the Q plus K method. You can also give your own kinship matrix. So if you have a kinship matrix that you want to use, that you calculate in some special way, you can put it in there as an argument, and it will realize, oh, this is a matrix. So it will use that instead. As long as it has the right shape, it will use it in the model. And as I mentioned, you can also just give some kind of population vector with a Q. I don't recommend to do this. I tried it, and it didn't work very well for, at least for the data that we were using. But if you want, you can. Yeah, one thing I should mention is this bin size parameter. So when you calculate the kinship matrix, what we do is we sample markers across bins of specified distance to have a homogeneous sampling over the whole genome, even if your markers are very heterogeneously distributed. So the size of the bins are defined by this parameter. If you give a map in centimorgans, that this bin size should be a number in centimorgans. You can use somewhere between 1 and 0.1. Gives okay results in my experience. If you use base pairs, then give bin size in base pairs. So something like 1,000 base pairs or 10,000 base pairs, 100,000, doesn't matter. Yeah, that's what I just said right now. Yeah, there's other functionalities to map-QTL, which I don't think I should cover right now because it's a bit long otherwise. Basically, you can impute missing values using a very simple imputation if you have missing values in your genotypes. You can add cofactors, either categorical or numeric. If you're interested in this, have a look at the vignette. And you can also calculate the significance threshold either using a permutation approach, which takes forever, but if you really want to be extremely accurate, that's the best method. Or you can do an estimation with the Lee and G method, which basically just checks the number of effective independent tests and then gives you an estimate. This works pretty well, and it's much faster, in my experience. So yeah, that's about it for the mapping functionality. I see that I'm already at 9:30 for me, so probably 2:30 for you guys. So yeah, I guess that's the end of my presentation. But if there's any questions, you can ask me now, or you can email me. My contact is also in the GitHub page. So feel free to email me about questions, or if you're interested to use it, or if you have examples or things that don't work, feel free to message me.
Thank you very much, Alejandro.